Hi, welcome to the lecture on histology, which is the study of the tissues that make up the body. Here on this slide, we have a picture of brain tissue. So the black dots that you see are actually neurons, and the little tree-like things that come out of them are their dendrites, which are the processes they receive signals on. This is a Golgi stain, if you're interested to know that. Okay, so every organ in the body is composed of 4 different tissue types. You only have 4 basic types of tissue in the body. They are epithelial tissues, connective tissues, and then muscle tissue and nervous tissue. We're mostly going to cover the epithelial and connective tissues, and then muscle tissue and nervous tissue each have their own chapter later in this mini-mester. Epithelial tissues cover surfaces in the body, so wherever you have a surface, obviously on the outside of your body, but also internally, like the interior of your blood vessels, that's a surface. Also, like the lining of your gastrointestinal tract. So in all those places, you'll find epithelial tissue. Every tissue type is divided into extracellular matrix and cells. So epithelial tissue has little extracellular matrix, and it's mostly made of cells. Extracellular matrix being basically everything that is outside of the cell. So that would include like liquid, but also molecules and protein fibers and stuff like that. Connective tissues connect, of course. They sort of support other tissues, hold things together, and also form just the basic body of a number of organs. They are mostly composed of extracellular matrix with few cells. Then you have muscle tissue, which is a special type of tissue because it is able to contract, and because muscle cells can carry electrical signals. Muscle tissue has little extracellular matrix. It's virtually entirely made of cells. And then finally, nervous tissue, which is also special. It can also carry electrical signals, and it can send messages and process information. Nervous tissue is more of an even mix between cells and extracellular matrix. Before we get into the details of those different tissue types and their subtypes, we're going to talk about what exactly is in the extracellular matrix and also junctions between cells. Then we'll switch to the different epithelial and connective tissue subtypes that we have. Okay, so in your extracellular matrix, you have 2 basic types of things. You have the ground substance and the protein fibers. Protein fibers are larger fibers. They're sometimes extremely long, that run through the extracellular matrix. And then ground substance is everything that's kind of smaller or flimsier than a protein fiber. So your ground substance is going to be divided into the small stuff and the big stuff, but even the big stuff is not crazy huge. Your small stuff is including water, various ions, nutrients like glucose or ATP, or vitamins, and more. So for instance, in nervous tissue, you would also have a lot of signaling molecules in the small stuff of the ground substance. Then there's 3 basic types of larger molecules in ground substance. You have glycosaminoglycans, abbreviated GAGs, or GAGs. You have proteoglycans, and you have glycoproteins. Which we'll get into what those are exactly in a moment. Then your protein fibers, you have 3 basic types of protein fibers that would be found in extracellular matrix. Those are collagen fibers, elastic fibers, and reticular fibers. Okay, so glycosaminoglycans first, or the GAGs or GAGs. So that's a big long word that probably sounds very confusing. But if you break it down into its parts, it actually tells you what it is. So it's a type of polysaccharide. Poly means many, and saccharide means sugar. So polysaccharide means many sugars. It's just a long chain that is made out of sugar. A glycan is also a long chain of sugars. So a glycosaminoglycan that has the word glycan at the end there, that is a type of glycan, so it is a long chain of sugars. Polysaccharide and glycan are basically synonyms. The glycosamino is telling you what type of sugar is making up that chain. So for glycosaminoglycans, every other sugar in the chain is a glycosamine, and then the others are just regular sugars. If you break down the word glycosamine, glyco means sweet, so that means it's a sugar. Amine is a functional group in chemistry that contains a nitrogen atom. It's a NH2 group. So a glycosamine is a sugar that has an amine group, or a sugar that has a nitrogen atom in it. So as an example here, here we have glucose, and then right next to it here, right here, you have your glucosamine, which is the glycosamine version of glucose. And you can see it's exactly the same molecule as regular glucose. The only difference is that it contains this NH2, which is the amine part of the sugar. So in your glycosaminoglycans, you basically have a long chain of sugars in which every other sugar has a nitrogen atom in it, like this glucosamine. Okay, so at the bottom of the slide here, you have an example of a common glycosaminoglycan. This is hyaluronic acid. It's one of the larger glycosaminoglycans that you have. So it's just these two different sugars. This one being your normal, this one being your normal sugar, and this one being your glycosamine. There you go. There's your nitrogen right there in the glycosamine. And then these two units just repeat over and over again to make a very long chain. You may be familiar with hyaluronic acid. It's sold in certain skincare products. This glycosaminoglycan, as well as all other glycosaminoglycans, are going to have these OH groups. For instance, this one right there, near the double bonded oxygen. If it's actually in tissue, this hydrogen on that OH group is going to dissociate and go away to join the small stuff in the ground substance as an ion, which would leave just a lone oxygen here that would have a negative charge on it. So in tissue, your glycosaminoglycans are always having these oxygens that have negative charges. So they are negatively charged molecules. And remember that this whole thing right here, that's just one part of the hyaluronic acid molecule, which would repeat many times. And every time it does, you're gaining one more negative charge right here on this oxygen, which means the entire molecule has a substantial negative charge on it. And that charge is important for the function of the glycosaminoglycans, as we are about to see. Okay, so you have lots of different types of glycosaminoglycans. They're always a long chain of sugars, where every other sugar is a glycosamine, so just a sugar with a nitrogen atom. But some of them are just somewhat long, and some of them are extremely long. Like the hyaluronic acid is one of the ones that's very long. But they all have a negative charge. Because they have that negative charge, they're going to attract anything in the ground substance that has a positive charge, but they're also going to attract water and polar molecules. So I don't know if you may remember from chemistry, but water is polar. You can see the structure of water on the slide here. And it's just an oxygen with two hydrogens bonded to it. but the electrons are not shared equally in those bonds between the oxygen and the hydrogen. The electrons are actually being pulled towards the oxygen. Oxygen is very electronegative, which just means that it attracts electrons to itself very strongly. It's one of the most electronegative elements, actually, so it attracts electrons more strongly than almost any other element can do. So whenever it participates in a bond with hydrogen, the electrons that are in that bond are going to spend most of their time near the oxygen, and they won't spend very much time near the hydrogen. So the oxygen is kind of hogging those electrons. Therefore, you get this sort of uneven charge distribution on the whole molecule. Since most of the electrons are around the oxygen, that means that most of the negative charge that they carry is around the oxygen, which leaves less negative charge over by the hydrogens. So the hydrogens are going to have a partial positive charge, or the end of the molecule where you have those hydrogens is going to have a partial positive charge, and the end of the molecule that is by the oxygen is going to have a partial negative charge. So that is the basis for the polarity of water. Any compound that has the electrons shared unequally is going to be polar. It's going to have one side being partially negatively charged that has more electrons, and then the other side that has less electrons is going to be partially positively charged. Any polar molecule is going to be attracted to any ion. So for water, that's going to be attracted to the negative charge on your glycosaminoglycans. Specifically, the end of the molecule that has the hydrogens on it is going to be attracted to the negatively charged oxygen on your glycosaminoglycans. So that means that when you have glycosaminoglycans in your ground substance in the extracellular matrix, they're going to be attracting water to them, also all other polar compounds that may be there, also positive ions like sodium, but most importantly, that water. So that's going to help the tissue maintain its water content. And when you have a lot of water in the extracellular matrix, because you have all of those glycosaminoglycans, that makes the tissue very firm and hard to compress. So that's why you see hyaluronic acid being sold as a skincare product. The idea is that it would attract water in the extracellular matrix of your skin and, you know, keep your skin plump and firm and youthful looking, you know, puff out any wrinkles, I suppose. Whether or not it works like that, that I don't know. The next type of large molecule in your ground substance is the proteoglycans. So a proteoglycan is a sugar chain with some protein attached to it. Glycan would be the base word in proteoglycan, and a glycan is again a long chain of sugars. And then proteo, meaning proteins, that's the prefix. So proteoglycan must be a long chain of sugars with some protein attached to it. So it's mostly sugar with a little bit of protein stuck on. The sugar part of the proteoglycan is in fact a glycosaminoglycan, or a GAG. It's essentially a core of protein with glycosaminoglycans coming off of it. They're kind of coming off in all directions, as shown by your illustration here. Like this in the box here, that is a proteoglycan. So the long core there is a sugar, and the little things coming off are your, oh sorry, the long core is a protein, and the little things coming off are your sugars, your glycosaminoglycan sugars, coming off in all directions to make sort of like a bottle brush shape. And then it's possible to have those either sort of separate and on their own in the tissue, these individual proteoglycans. But it is also possible to have them attach themselves to a larger glycosaminoglycan in what would be called a proteoglycan aggregate. So here, here this large, long green molecule, that is hyaluronic acid, glycosaminoglycan, and then coming off of it at different points, like right there, right here, right here, right here, you have individual proteoglycans. So again, a protein core coming off with smaller glycosaminoglycans coming off of that. So a proteoglycan aggregate would be like a long string with these little bottle brushes coming off of it on all sides. By the way, these larger pink fibers, those are the actual protein fibers. So you can see that their diameter is significantly larger than the glycosaminoglycans. The longest of the glycosaminoglycans, the hyaluronic acid, could be comparable in size to a protein fiber, in length anyways, but can never begin to match it in girth. Okay. So, since proteoglycans contain glycosaminoglycans, they also have that negative charge on them, so they're also going to have all the same roles in the tissue that glycosaminoglycans would have. Basically, they're attracting water, ions, other, well, positively charged ions, other polar compounds, anything like that, but primarily water. Then the physical shape of the proteoglycan with the bottle brush type shape helps to trap stuff. So it could trap like large molecules in the tissue, but also microbes. And it's not going to exactly like trap them and hold them firmly in place, but it can like deter their movement through the tissue. So if you had, you know, some microbes that got into like a wound that you had, and they were trying to migrate or move through your extracellular matrix to get to other tissues or to your bloodstream, if you had a lot of proteoglycans in that tissue that they were in, the movement of the microbes would be slowed down by having to get through all those bottle brushes. And the same would hold true for cancer cells. To have like a really dangerous cancer, the cells in that tumor need to detach from the original tumor and migrate through the tissue to a new location. That would be part of the process called metastasis, which is, you know, sort of the hallmark of a late stage, very dangerous cancer. As your cancer cells are trying to move through the tissue, they have to get through all those proteoglycan bottle brushes, which kind of slow their movement. So for cancer that's really good at you know, metastasizing, or you know, has metastasized a lot, you're probably going to see that those cancer cells have a way of degrading the proteoglycans to help them move through the tissue more easily. Then you have glycoproteins, finally, which is sort of the opposite of a proteoglycan. For proteoglycans, the base word is glycan, which is the type of sugar, and then the prefix is proteo, so it's a sugar with some protein. Glycoproteins are the other way around. The base word is protein, so it's mostly a protein, and then it has the prefix glyco, meaning sugar. So it's a protein with a little bit of sugar stuck onto it. So you can see in the example here, this is your protein part, and then this would be your little bit of sugar stuck on. You can have, like, there's many different glycoproteins. They have a great diversity inside the cell and outside the cell. If you read the textbook says that glycoproteins in the ground substance are all anchoring cells to the extracellular matrix. They're all cellular adhesion molecules, and that's only one type of glycoprotein actually. So inside the cell you have enzymes and receptors and hormones that are glycoproteins. It's very common. Also outside the cell in the extracellular matrix, you can have glycoproteins making up part of like some protein fibers. Yeah, so it's a class of molecule that has a great diversity and is found in many locations. But for our purposes, the type of glycoprotein in the ground substance of the extracellular matrix that you would want to remember is the cellular adhesion molecule. Right. So those are basically just linking. They're going to be attached to the membranes of cells in the tissue, and then the other end of that glycoprotein would be attached to a protein fiber running through the tissue, usually a collagen fiber, which is the fiber that gives strength. Right, so the role of the glycoprotein cellular adhesion molecule is going to be to attach cells to the extracellular matrix, which anchors the cells in place. It prevents them from moving around. It helps the tissue maintain its organization. If you have metastatic cancer, in order to have cancer and become metastatic, the cancer cells have to start expressing an enzyme that will allow them to degrade cellular adhesion molecules so they can move through the extracellular matrix. If they don't have an enzyme to degrade the cellular adhesion molecules, it's impossible for that cancer to metastasize. So it's a necessary step in the development of metastatic cancer. Right, so here's an image kind of illustrating the difference between glycoproteins and proteoglycans. So you can see for the glycoprotein, most of the molecule is the protein bit, and then you have some little bits of sugar coming off. For the proteoglycan, most of the molecule is that large protein core there with the little bits of glycosaminoglycans coming off of it to make the bottle brush. Okay, now for our protein fibers. The first type is the collagen fiber. That's the type of protein fiber that gives strength to the extracellular matrix and to the tissue. Collagen fibers have a structure that's sort of similar to like a steel rope or a steel cable, where you have one like long fiber of steel and several like this kind of thin, and you get several of those together and wind them up to make a fatter fiber, and then you get several of those together and wind them up, and that makes your whole steel rope. So you have kind of fibers, different levels of fibers entwined within each other. It's similar for collagen. You start out with an actual chain of amino acids. If you get 3 of those amino acid chains together and wind them around each other, you have a tropocollagen that's considered like the base molecule for a collagen fiber. So you can see in the blue, red, and black there the 3 different amino acid chains that are wound together to make one of those. And then if you take like several of those tropocollagens and wind them together, you can get a microfibril. So one of these right here would be a microfibril. If you get several of those microfibrils together and wind them up, you'll have a fibril. Those microfibrils within the fibril are also cross-linked to each other. So you can see these little cross-links between the microfibrils there. That's also helping to give strength to the overall fiber. And then finally, if you take several fibrils and wind them together, then you get finally your final collagen fiber, which has like it has like 4 different layers of things that are all wound together to make the final fiber. So you don't need to know that precise structure, just that it has those different layers of like a hierarchy, I guess, of fibers of different thicknesses wound together. Each one itself made up of smaller fibers also wound together. So that structure makes collagen very resistant to stretching forces, also known as tension. Makes it a very strong fiber that it's hard to stretch. You have actually a lot of different types of collagen fibers depending on how you actually arrange the tropocollagen molecules. You can get different fibers out of it, but collagen 1 is by far the most common type of collagen fiber. In fact, it's the most common type of protein fiber overall, and one of the most common proteins in your whole body. Collagen accounts for 20 to 25% of all of the protein in your body, well, collagen 1 specifically. Right, so here's a different illustration of the structure of collagen. So you can see the different fibers here in here would be your amino acids, 3 wound together to make a tropocollagen here, wind those together to make a microfibril, wind those together to make a fibril, and then wind those together to make your whole fiber there. Right, so so you can see that would probably give a lot of strength, similar to how it does for the steel rope. So the role of collagen fibers in tissue is going to be to make the tissue strong, but specifically it's going to make the tissue able to resist stretching forces. So collagen will bend very easily, but it does not stretch. Right, so the specific type of strength that it confers on the tissue is resistance to tension, which is resistance to stretching forces. Okay, next you have your elastic fibers. Elastic fibers consist of elastin with microfibrils. So the microfibrils are tiny little protein fibers. There's lots of different types of them. You can see them here in the illustration of an elastic fiber. They're kind of they're all like connected to each other, and they're kind of around the outside of the fiber. The role of those microfibrils is just to give the fiber sort of shape and structure, make it look like it is a fiber. Then inside of your microfibril outer layer, outer shell, you have the elastin. It's sort of this amorphous jelly-like mass filling up the interior of the fiber. The elastin, if you actually look at the molecular structure of it, it looks similar to this. Right, so you can see the elastin fibers themselves are these little curlicue spiral type shapes. They have all different types of shapes, but they're all curled up or curlicued. And then between them, you've got these crosslinks. So they're all linked to each other, and they all have these different curlicue shapes. So then what's going to happen if you were to stretch an elastic fiber? The elastin within it would stretch. All of those little molecules of tropoelastin are going to stretch out to be kind of straight. They all are still cross-linked to each other, right? So they're going to remain attached, but since they're all stretched flat, the whole length of the fiber is going to get much longer, and then if you let go, they bounce back to their original curly shape. So that's the way they're going to provide elasticity to the tissue, or to the fiber and then to the tissue. Right, and of course, as the name pretty well indicates, the role of elastic fibers in the tissue is to provide elasticity, which means that if the tissue is stretched, it will bounce back to its original shape. Finally, you have the reticular fibers. Reticulum is Latin for a little net, so if you ever see the word reticule in biology, that usually means like a net. So reticular fibers are fibers that are like a net. Reticular fibers are actually a type of collagen. They are collagen 3, but of course they discovered the fiber long before they knew, about the precise structure of the fibers. So that's why they're called reticular fibers. They were named, more for their function in the tissue than for their molecular structure. Right, but unlike collagen 1, which has a lot of tropocollagens wound together to make microfibrils, microfibrils wound together to make fibrils, fibrils wound together to finally make fibers. In reticular fibers, you just have one chain of tropocollagens, so it's way thinner. and shorter and weaker than collagen 1. It does not really provide strength to the tissue. It's not strong at all. But these microfibrils, seen right here, of the collagen 3 triple collagen, those microfibrils are going to be arranged in all directions like a net. So you might have one, they might be arranged kind of like so, and all attached to each other. So wherever they cross, you would have little attachments between them, like little cross links, like so. Right, so they're sort of arranged in a shape that's like a net. The role of reticular fibers in the extracellular matrix is to provide support for delicate structures and also to help trap particles and especially microbes. So one of the prominent places to find many reticular fibers would be in tissue of the lymphatic system, which is prominently associated with the immune system. It has a couple of different functions which you will learn about in A&P 2. But one of the primary functions is with regard to immunity. So for instance, your spleen is an organ in the lymphatic system. Its job is to filter your blood, to filter microbes out of your blood if they happen to get into your blood, which, of course they shouldn't in the first place, but sometimes things don't work out the way they should. So if you have microbes in your blood, then when your blood is filtered through the spleen, you have a lot of reticular fibers in the extracellular matrix of the spleen. So as the microbes are traveling through that tissue, they're going to be trapped in those little nets formed by the reticular fibers. And then what else you have in the spleen is a lot of immune cells. So the microbes are going to be trapped there, and then the immune cells can come in and kind of respond to the microbes and deal with them. So here's a summary of our components of the extracellular matrix. You have the ground substance and the protein fibers. The ground substance being sort of the small things in the foundation of the extracellular matrix, and the protein fibers being larger fibers that run through it. So in the ground substance, you of course have your small stuff, water, ions, nutrients, stuff like that. But you also have your larger molecules, which would be the glycosaminoglycans, proteoglycans, and glycoproteins. The glycosaminoglycans and proteoglycans both have a role in maintaining the water content of the extracellular matrix because they have a negative charge, so they attract water. And then when you have a lot of water in the extracellular matrix, that maintains the firmness of the tissue and helps it resist compression. Proteoglycans, because of their bottle brush shape, especially in the aggregates, are also able to trap microbes, or at least delay the movement of microbes and cancer cells through the tissue. Right, and then proteoglycans, right, remember they consist of a protein core with little glycosaminoglycans coming off, which is why the proteoglycans are also negatively charged, just like the glycosaminoglycans. Then finally, you have the cellular adhesion molecules, which are a type of glycoprotein. The role of those is going to be to hold cells in place by binding cells to fibers in the extracellular matrix, so that anchors the cells and helps the tissue maintain its shape and organization. Then for your protein fibers, most prominently you have collagen, especially the collagen 1 fibers. Those are just providing strength, specifically allowing the tissue to resist tension, which would be stretching forces, so helping the tissue resist stretch. The structure of collagen would be lots of little fibers wound together to make a bigger fiber, then those ones wound together to make a bigger fiber, and so on. So a structure that's similar to a steel cable. Then you have elastic fibers, which of course provide elasticity, because they have all those in the elastin within that fiber. You have all those little curlicues that are attached to each other with crosslinks, so they can, if the tissue is stretched, they will straighten out and flatten out, and then when the stretching force goes away, they will bounce back to their original curly shape. Then your reticular fibers, which is actually a collagen 3 fiber. Those are supporting fine structures and trapping microbes, or mainly microbes actually. Potentially maybe cancer cells, depending on where the cancer would be. Right, and that's because of the structure of the reticular fibers. They are all kind of arranged in different directions. So they're all running in different directions through the tissue, and they're all cross-linked to each other. So they're basically physically forming a net in the tissue. Oops. Okay, yeah, so now we have our cell junctions, which are just connections between cells. You have three basic types of cell junctions: tight junctions, desmosomes, and gap junctions. Right, so tight junctions look sort of like zippers, sort of like zipping two cells together, the walls of two cells together. So you're going to have these lines of proteins, which are integral membrane proteins. That just means that the protein is embedded in the membrane of the cell. You're going to have lines of those on each cell that's going to participate in the junction, and then those integral proteins just meet up and bind together, thereby binding the two walls of the cells together. And you're usually going to have multiple lines of them like this. The point of that is that it just makes a very tight connection between the two cells, so it's very difficult for a solute, for a molecule to move between here. The idea is that like not even water could get past here. The cells are so closely held together. Depending on how many lines of these tight junctions you have, that would determine like if a tiny bit of stuff can get through or if literally nothing could get through. Right, so the point of those junctions in the tissue would be to maintain barriers so that you can control how things are crossing that barrier. If nothing can get between the cells, that means that for something to cross the barrier that those cells are forming, it would have to be taken up into the cells. So the cells are going to be able to control what they take up and cross. So an example would be like lining your gut. You don't want things to be just sort of getting past those cells that line your gut willy-nilly. You want to specifically take things across that you want, like nutrients, but you don't want like toxins, of course, to get into your body from your gut. So you could have tight junctions there to maintain like a strong barrier. Also, in the brain, you have the blood-brain barrier. So you have to carefully protect your brain from toxins and different chemicals that could be in your blood, but also hormones in your blood. A lot of hormones in your blood are actually signaling molecules in the brain, and if they got into your brain from the blood, that would create kind of like chaos, and your brain wouldn't be able to process information properly. So you have a blood-brain barrier that prevents substances from just leaking from your blood into your brain. So the cells lining those blood vessels in your brain are joined together with tight junctions so that nothing can get between them. So if something is going to get into your brain from the blood, it has to be specifically selected by those cells lining the blood vessels and transported across the cell into the brain. So your cells are going to be specifically transporting glucose into your brain, for instance, but they wouldn't transport a hormone into your brain. Right, that also just helps maintain polarity. So a polarity is a difference in the environment on one side of the cell versus the other side of the cell. Right, so anywhere you need to maintain polarity, tight junctions are helpful because they kind of isolate the two sides from each other, so that so that no chemicals can just kind of diffuse from one side to the other. Then you have your desmosomes. The role of the role of desmosomes is to bind cells together. Desmos is a Greek word that means bond. Soma is also Greek, means body. So desmosomes just means bonded bodies, literally. And again, of course, you don't need to know like the complicated structure of the desmosome, but I'll just briefly go over it. So the basis of your desmosome would be this, let's see, these plaques. Yeah, so these red plates embedded in each cell's membrane, those are plaques. So that includes proteins, sort of structural proteins, that are just giving strength to the junction. And then coming out on the extracellular side, so pointing outside the cells, you have glycoproteins on each side, lots of glycoproteins sticking out, which then find the glycoproteins on the surface of the other cell and bind them. So the cells would actually be physically bound together with these glycoproteins. Whoops. Yeah. Then on the internal side, so facing into the cell, you have these filaments, and these are going to be bonded to the cytoskeleton of each cell. The cytoskeleton, if you remember from general biology, it's basically a network of proteins running through the cell that's like a scaffolding or a skeleton. Yeah, so it just provides structure and shape to the cell. So by having these desmosomes actually linked to the cytoskeleton with these filaments, you can basically, in function, you can link the cytoskeleton of one cell to the cell that's next to it, which provides a lot of strength. So desmosomes are going to be a very strong way to kind of hold cells together. The role of desmosomes in the tissue is to hold the cells together. It provides a lot of strength to the tissue. So if you remember that epithelial tissues don't really have extracellular matrix, that means they don't have like collagen fibers running through that would normally be providing strength to the tissue. So instead, they're going to get their strength prominently from desmosomes. And if you have an epithelial, a layer of epithelium, well, if you have epithelial tissue that has a number of layers of cells in it, you can have desmosomes, at all points throughout those layers, so that all the layers are connected to each other. So the cytoskeleton of each cell in that epithelium, as shown by these red fibers here, would be connected to the cytoskeleton of every other cell in a network that provides a lot of strength. It also helps distribute strains that are placed on the tissue. So if you put a strain on the tissue at a specific point, like right here, then that's going to be distributed. That's going to be distributed amongst the other cells that are connected by desmosomes. Ultimately, it would be distributed around the whole area of the tissue, which helps provide strength, of course. And then finally, we've got gap junctions. Gap junctions are made of also integral membrane proteins, so proteins embedded in the cell membrane, but in this case they're arranged to form sort of like a pore that stuff could like travel through. Right, so you have a pore on one side, on one cell, and then on the other side of that junction you have the same pore in the other cell, and they attach to each other to make a channel, so that the cytosol, or the fluid inside each cell, is connected. So if you have a small molecule like an ion, it can travel right through. It can travel right through that desmosome. It can travel right through that gap junction. If you're talking about ions that are passing through, like a sodium ion, for instance, that would be carrying its charge with it, and anytime you have electrical charges moving, that's a current. So gap junctions can carry electrical currents from one cell to the next cell if ions are passing through them. And some gap junctions can select what specific type of molecule they'll allow to pass through. So the role of gap junctions in the tissue is going to be to allow cells to communicate with each other and to synchronize their activity. So for instance, in your heart, all the muscle cells in the walls of your ventricles or the walls of your atria, they need to be able to contract together at the same time. If they don't all contract at the same time, you're not going to have enough force generated for the little heart to pump blood through your entire body. So they have to contract all at once. And the way they do that is by being connected to each other using gap junctions. So the gap junctions allow the muscle cells in your heart to synchronize with each other in their contractions. You also see it in the brain sometimes. So for instance, for milk ejection or lactation, there's certain cells in your brain, certain neurons that will join together using gap junctions so that they can fire simultaneously, which is necessary to allow the milk ejection. Also, you have a role for gap junctions in growth and development. It's not clear exactly what that role is, but it is very important. If you're not able to form gap junctions in your tissues as they're developing, you won't be able to develop correctly. So most of your cells would actually have expressed gap junctions at some point. And then, when you are developed enough that you don't need the gap junctions anymore, then they just are broken down and they go away. So as an adult, most of your cells would not have the gap junctions anymore. Okay, so now we can actually get into the types of tissues, starting with the types of epithelial tissues. Right, so as you remember, the epithelial tissues are covering surfaces in the body, and both internally and externally. There's a variety of functions that epithelial tissues can serve depending on the exact tissue that we're talking about and where it's located at. So some of them function in protection. So for instance, the skin has epithelial tissue lining it, of course, since it's covering the surface of your body. And one of its main roles is to protect your body from everything that's out there in the environment, and a lot of microbes. So you would really be having a trouble keeping microbes out if you didn't have your skin protecting you. And it is the epithelium on the skin that is the first and primary barrier between you and the environment. So that's also an important role in immunity that's played by the skin. But also sort of internal surfaces, it can have a role in protection when epithelial tissues coat your internal surfaces as well. So for instance, the epithelial tissues that line your gastrointestinal tract are kind of protecting you from, all the things that are in your gastrointestinal tract, which includes, again, many microbes. Also, maybe sometimes some toxic things that you may have eaten. The gastrointestinal tract is exposed to the environment, so it's important to protect you from it, even as you're needing to absorb nutrients across it. Then you have secretion. Glands are always lined with epithelial tissue, so anytime you're secreting something, that could be like sweat on the surface of your skin. Internally, that could be like, say, the pancreas secreting digestive enzymes into your intestine to help you digest, or it could be the walls of your stomach secreting acid into your stomach to help you digest. And there's a lot of different places where you would secrete something. Glands are pretty much always just made with epithelial tissue, and it's those cells in the epithelial tissue that are actually making the substance and secreting it. Then you have transport, so that would include gas diffusion and absorption. In your gut, of course, you need to absorb nutrients from your food into your bloodstream, so it would be epithelial tissues that are handling that are specifically transporting nutrients from the gastrointestinal tract into your bloodstream. And then let's say in your lungs, you need to absorb gases from the environment. You need to absorb oxygen into your bloodstream, and as your tissues as your cells are metabolizing, they generate carbon dioxide as a waste product, and that needs to be exhaled. So that needs to travel from the bloodstream into the air in the lungs and leave your body as a waste. Right. So in the lungs, you need to exchange oxygen and carbon dioxide between the blood and the environment, and epithelial tissues allow that to happen. Finally, you will frequently have sensory receptors embedded in epithelium. So of course your skin is studded with touch receptors that are frequently located in the epithelial part of the skin. Also, you have in the skin receptors that are for temperature, so they let you know if it's warm or if it's cold. And you have nociceptors that let you know if there's pain. Nociceptors sense pain, so that could mean like physical damage to the skin, or extreme temperatures, or a bad chemical like maybe acid. All of those things could activate nociceptors in the skin and generate a sensation of pain. Also, in your deeper tissues, you frequently don't think about them as having sensation, but they generally do. It's just that you're usually not conscious of it. It's not. They aren't senses that you're consciously aware of usually. But in almost all organs that have a hollow passage in them, you would have stretched receptors embedded in the lining of that organ, frequently in the epithelium, right, that's actually coating all those internal surfaces. So for instance, in your gut, throughout your whole gut, you have stretch receptors embedded in that epithelium that let you know just how full you are. So if you feel really full, that's because you're sensing the activity of those stretch receptors. They're turned on anytime they're stretched. But you're always having that sensation, like your brain always has access to that information. You're just usually not consciously aware of it. Also in your blood vessels, in the largest blood vessels, so like in the aorta, for instance, you have stretch receptors embedded in the lining of that vessel, which will let you know, like you're not conscious of it, but they'll let your subconscious brain know how much stretch is in the vessel, which relates to what the blood pressure is. So that's how your brain is able to track what your blood pressure is, by using those stretch receptors. In general, the epithelium is always the interface between the body and the environment, although you also find it in other locations as well, so like lining your blood vessels, lining your kidneys, stuff like that. But anytime you're going to have an interface between the body and the environment, you're going to have epithelial tissue there. And so you have several common characteristics that all epithelial tissue subtypes have in common. So first, it's always located on surfaces. It always has connective tissue underneath it. You have no blood vessels in epithelial tissues. It's mostly just cells, wall to wall. You have very little extracellular matrix. So in general, you actually don't have any extracellular matrix to speak of between the cells. It's just cells like directly touching each other, nothing between them. At the bottom of the epithelial tissue, you have a little bit of extracellular matrix there, which forms the basement membrane. You also have polarity in polarity in epithelial tissues, which means that one side of a cell is different in some way from the other side in terms of its chemistry, well, like what molecules it has, or in terms of its function, generally both, in terms of its structure. So for epithelial cells, you can generally name the different sides. So they'll have an apical side, which is the top side, a basal side, which is the bottom side, and then lateral sides, which are the sides. The apical side would always be the side that is facing towards the surface, and then the basal side would be the side that's facing into the rest of the tissue. Then you have a basement membrane that is found at the bottom of your epithelial tissue, at the bottom of every epithelial tissue. That is the only extracellular matrix in that tissue to speak of. It's made of extracellular matrix. The top layer is actually made by the epithelial cells, and then the bottom layer is made by the connective tissue. Right, so on my slide here, basal lamina literally means bottom layer, but it is actually the top layer. It's beneath all the cells, but it's above like the connective tissue underneath. Okay, so here you go. Here's an illustration of some epithelial tissue. So these would be your epithelial cells here. You can see they have some junctions between them. Here you have, let's see. Okay. Here you have tight junctions to control the movement of substances across that barrier formed by the tissue. Here you have desmosomes connecting the cells together, giving strength. Here's your apical surface on the top here, and the basal surface would be, on the opposite side. This is your basement membrane down here. This part, this top layer, is made by the epithelial tissue itself, and then the layer underneath that, this layer, this is made by the connective tissue. As you notice, you have no blood vessels in your epithelial tissue. All of the blood vessels, all of your blood vessels are in the connective tissue that is underneath. Okay, so here's the ways that we categorize epithelial tissue. You have two different ways of categorizing it, either by thickness or by cell shape. So by thickness, you can either have one layer of cells, which would be a simple epithelium. That's better for transporting substances across it. Or you could have multiple layers of cells. That would be a stratified epithelium, which is stronger and better for protection. Then you have several, three different cell shapes. You have squamous cells, which are flat. You have cuboidal cells, which actually in the actual tissue don't really look like hexagons or like cubes, but they are generally about as wide as they are tall. So usually the nucleus is kind of centered in the middle of the cell. That would be the way to recognize it. Then you have columnar cells, which are always taller than they are wide. And the nucleus would be located more towards the basal surface of the cell, so the nucleus is on the bottom. Then if you combine these two different ways of categorizing the epithelial tissue, you can get all the actual subtypes of epithelial tissue. So we have simple squamous, stratified squamous, simple cuboidal, stratified cuboidal, simple columnar, stratified columnar. Those are all subtypes of epithelial tissues. And then we have also a couple of special subtypes as well. So we'll go through those subtypes one by one, starting now. Right, so first you have simple squamous, a single layer of flat cells. This is an extremely thin type of tissue, so it's pretty difficult to see in the microscope. It doesn't generally look as clear as the image here on this slide. Let's see, this side would be an actual microscope image, and this side is a drawing based on that image, but it usually actually does not look quite that clear. So since this is the thinnest layer of epithelial tissue that you can have, it's extremely good at diffusion. It's also very weak. So it's both the best at diffusion and the weakest. So in general, you would not find it in a place where you have a lot of mechanical stresses because it's delicate, but you would find it anywhere you need fast diffusion across the epithelium. So, for example, you're going to find simple squamous tissue lining the alveoli, which are the tiny air sacs in the lungs where gas is actually exchanged. That would be, that's actually not what's seen in the image. Yeah, so that in those alveoli, like you're having oxygen cross into the blood and carbon dioxide cross from the blood into the air. And yeah, so for those gases to cross well, you need a very thin layer of cells. Also, some tubules in the kidneys where the urine is initially being filtered out of the blood and formed, you have simple squamous there. Also lining your blood vessels. So frequently you actually do have mechanical stress in your blood vessels because of your blood pressure, especially in the arteries. But you still need to have simple squamous epithelium because, again, you have to be able to exchange oxygen well with the tissues and get carbon dioxide out of those tissues into the blood. You have to, and also glucose needs to leave the blood and enter the tissues, and other nutrients as well. So things really need to be able to get in and out of the blood easily. So you're always going to have simple squamous lining blood vessels. In your larger blood vessels that are exposed to more pressure, more mechanical stress, the strength of the vessel is going to be coming from other components of the lining, not from the epithelium. Finally, you have simple squamous tissue lining the serous membranes, which basically reduce friction in different parts of the body and kind of help create body cavities. So, for instance, right, the image there is actually coming from the serous membrane. Right, this image here is actually coming from the serous membrane surrounding the lungs that allows the lungs to move without rubbing against the cavity that they're located in, which would, be quite painful and would degrade the tissue. Next we have simple cuboidal. That is a single layer of cube-like cells, so cells that are roughly as wide as they are tall. It's also very good at diffusion because it's still a thin layer of cells, but it's not as good at diffusion as simple squamous, and it's also a little bit stronger than simple squamous. Since the cells are taller, you can have more desmosomes between them, giving them more strength. So you're going to find simple cuboidal lining surfaces wherever you still need to have good diffusion and absorption, but where you expect to have a little bit more stress occurring on the tissue in general. So that could be, for instance, if the fluid is going to be under some amount of pressure there. So you can find simple cuboidal lining kidney tubules. That's the example that's pictured there, right? So this epithelium right here is your simple cuboidal. Whoops. And it is lining a kidney tubule, so you would have urine forming in the empty space right here. And you need to have water able to move in and out of the forming urine, and ions and other substances be able to cross so that you can get the concentration of urine just right before it's sent out into the bladder. You also find simple cuboidal lining the alveolar ducts and the bronchioles. Those are the little passages in the lungs that lead directly into the alveoli. Right, so since they're a little larger, they're going to have the simple cuboidal, but that's not the primary site of gas exchange, which would be the alveoli where you have your simple squamous. Finally, you can frequently find simple cuboidal lining the ducts of various glands. Next, you have simple columnar, which is a single layer of columnar cells. So you can see in the image here that the nuclei are more towards the bottom of that epithelium, which is a characteristic of simple columnar tissue, or columnar cells in general. So simple columnar tissue is still just one layer of cells, so it is still pretty good at diffusion and absorption, but it's not nearly as good as simple cuboidal or simple squamous. And it's even stronger still than those other simple tissue types. On the apical surface of those cells, which would be the surface here, on the apical surface, you're frequently going to have either microvilli or cilia. Microvilli would be sort of like little projections, little bumps coming off the top of those cells, which increase the surface area of the top of the cell for absorption. So anywhere that you need good absorption, so for instance in the gut, you're going to have microvilli increasing surface area for that. On the other hand, you could have cilia located on that apical surface, which are sort of little whips basically that beat and move stuff along the surface. So for instance, in your lungs, in the bronchi, which are the larger passages in your lungs, you would find those lined with simple columnar epithelial tissue that has cilia on it. And if you happen to breathe in some particles, they would generally get stuck in the mucus that lines those passages, or that coats those passages, the bronchi. And then the cilia on those cells is going to move the mucus with the particles up and out of your lungs. That's called the ciliary elevator, and it's the way that you get particles out of your lungs. So of course, if a particle would be breathed in more deeply into the parts of your lungs that are lined with simple cuboidal or simple squamous, those types of tissue do not have cilia, so you can't have any way to move that particle out. So if you breathe a particle into the deepest parts of your lungs, it will be there forever. Don't smoke. As for the microvilli, you would find simple cuboidal cells with microvilli lining your stomach, your small intestine, your gallbladder, and some kidney tubules. You can find other ciliated simple columnar cells lining the uterine tubes, so that also known as the fallopian tubes, so that as you have like a fertilized egg there, it'll be moved from the ovaries through those tubes into the uterus. That way it can implant in the uterus where the baby would need to develop that. You also would sometimes find a simple columnar epithelium that does not have microvilli or cilia lining some of your glands. Okay, so here's a slide illustrating the difference between microvilli and cilia. On this side you have microvilli. You can see they're like tiny little bumps or tiny little nubbins poking out of the surface of the cell, increasing surface area. And on the other side you have your cilia. So those are a good deal longer and move in kind of a whip-like motion to move things along. Okay, finally, you have a special type of simple epithelium, which is pseudostratified columnar epithelium. It is one layer of columnar cells, but the cells are packed so closely together that it looks like it's multiple layers. So you're usually going to find it looking somewhat like this, where you have nuclei at different layers, right? So you have like a layer of nuclei right here, another one right here, and then one at the very bottom. So it looks like there's multiple layers of cells there, but actually it's all one layer. But some of the nuclei are pushed up above the others because the cells are so tightly packed. So with this type of epithelium, the cells are so close together that diffusion is pretty difficult. It's technically still possible, but you wouldn't use this type of epithelium anywhere you really need diffusion or absorption to occur. But it is much more protective because the cells are so tightly packed. So that's out of the simple types of epithelium, it's the worst at diffusion and absorption, but it's the best at protection. Frequently, those cells are going to have cilia on them, and a lot of the time they're also going to have, well, the tissue frequently will also have these these special cells in here that make mucus. So pseudostratified columnar epithelium is frequently going to be coated with mucus and then having cilia. The places to find it would be the largest bronchi in your lungs and then the trachea, which is your main airway there, leading out to the mouth, well, to the throat and then the mouth. Also inside of your nose, you'll find the pseudostratified columnar epithelium. So those are surfaces that are in more direct contact with the environment. They need more protection. You're going to have mucus there to trap any microbes or other particles that come in, and then cilia to move it up and out. Okay, now for our stratified types of epithelium. First you have stratified squamous, multiple layers of flat cells. Actually, the bottom layers look way closer to being cuboidal, and the middle layers are sort of amorphous, so they have weird irregular shapes. But you're actually classifying the type of cell by shape based on the top layers. So in these topmost layers right here, the cells are flat, and that's why it's considered squamous, stratified squamous. For stratified squamous, the bottom layers of cells down here, specifically, like especially the bottommost layer, but also like 1 or 2 layers above it, those cells are actively dividing, which is unusual for cells in the body of an adult. And so as cells get born, they're kind of pushed up through the epithelium. Eventually they reach closer to the top. So as they get to the top, they're frequently going to be dying because there's no blood vessels in epithelial tissue. So the further they get to the top, the further they are from the nearest source of blood, which would generally be, well, always be down in the connective tissue underneath the epithelium. So that's a far distance for oxygen to diffuse through the tissue, and at the top layer, these cells are going to be not getting enough oxygen, so they're going to die and slough off. So your stratified squamous epithelial tissues are continually being renewed in that way, where cells are newly dividing at the bottom, getting pushed up, and dying as they reach the top and falling off. You have 2 subtypes of stratified squamous epithelial. You have keratinized stratified squamous epithelium, and you have non-keratinized stratified squamous epithelium. In the keratinized version, the top layers are always dead, and the cells are all filled with keratin, which is a very hard protein. The only place where you can find keratinized stratified squamous epithelium will be in the skin. The skin is keratinized stratified squamous epithelium in its epithelial layer, which again means all those cells are filled up with keratin, which is a very hard protein. So when you combine that keratin with the multiple layers of cells that are there, all connected by desmosomes, you get a lot of strength out of the epithelium for being epithelium. So that is keratinized stratified squamous epithelium is the strongest type of epithelium. Non-keratinized stratified squamous epithelium is the same, but with no keratin. So you're going to find that internally. So for instance, lining the mouth, the esophagus, the vagina, and the anus. Those are places where you have a lot of contact with the environment. You can have a lot of mechanical stresses there. So you need good protection, but of course, since it's internal to the body, you also need to have moisture. Those should always be moist surfaces, which is why a non-keratinized epithelium is more appropriate. Okay, so here's an example of the skin and the layers of the skin. You won't actually need to know these different layers of the sorry, this is only the epidermis of the skin, which is the epithelial layer. You won't need to know the different layers within the epithelial layer of the skin, but here they are. So on the bottom, your cells here are actively dividing. Then as they go up, they're getting further from oxygen. They're also starting to fill up with keratin when they reach about this layer here. they're beginning to die. They're also being coated with lipid substances, that keep your skin water resistant, which is another reason why they die. So around here they die, and then in the top layers, all of these cells are dead. All of these cells are dead. But they're still getting pushed up as new cells are born from the bottom until they reach the very top layer and fall off to form the dust in your house. If you're looking at an actual microscope slide, it looks a bit different. At the top, it's pretty hard to tell. Oops. It's pretty hard to tell one cell from another at the top. They all kind of merge together. They're very flattened. The nucleus is usually gone because the cells are dead, and they're all filled up with keratin. Okay. And here's an example of your non-keratinized stratified squamous epithelium. Right, so you can see that your cells, also this bottom layer, those cells are dividing, and then as the cells go up, they flatten out, and closer to the top, they're actually dying, and they slough off and are replaced from below. Okay, now for our stratified types of epithelium, starting with the stratified cuboidal. It is 2 layers of cuboidal cells, sometimes 3. And the example image right there, it's kind of hard to tell how many layers there are, which is partly due to just the way the tissue is sectioned. It's probably sectioned at an angle there. Right, so it can be kind of hard to distinguish exactly how many layers are present, but ideally it should be just 2 to 3 layers of cuboidal cells in your stratified cuboidal epithelium, so it never gets like that thick. It's pretty uncommon in the body. You would find it lining certain glands, prominently your sweat glands and sebaceous glands, which are the glands in the skin that manufacture sebum, which is the oily substance that coats your skin to keep water in your body. You also find stratified cuboidal epithelium lining the ovaries and the testes. Then stratified columnar epithelium, which is 2 layers of columnar cells. So in general, if you find yourself looking at some columnar epithelium that looks like it has 3 or 4 layers, that's probably likely to be pseudostratified rather than just stratified. Right, because stratified columnar should always have just 2 layers of columnar cells. It's also very uncommon in the body. It's found lining certain glands, prominently your salivary glands, the conjunctiva, which is the membrane that covers the outside of your eye and also the inner surface of your eyelid. Also the male urethra is stratified columnar epithelium. Then finally, we have a special type of stratified epithelium, which is called transitional epithelium. It has multiple layers of cells. As you can see, it's kind of difficult to tell exactly how many layers of cells are there. They're a bit jumbled. But since you always go by the top cell in stratified epithelium to classify the cells based on shape, you can see the top layer of cells here doesn't look like any shape that we've heard about. The cells have these little domes on the top. And the role of this epithelium, or what makes it special, is that it can stretch well. So as you stretch it, if you were to stretch it, the cells on top would flatten out and they would start to look actually like squamous cells. But when you relieve the stretch, they come back to have that domed shape on top. So the dome is allowing that tissue to stretch. Therefore, you're going to find it lining most of your urinary system, except for the kidney tubules where urine is being formed actually, but wherever you would have formed urine passing through, you're going to usually find transitional epithelium. So that would be lining the ureter, lining the bladder, lining their urethra, except in males who have stratified columnar epithelium lining their urethra. Okay. So those were the subtypes of epithelial tissue. Now I'll very briefly mention glands, which are always composed of epithelial tissue. Glands make stuff and secrete it. You have two basic types of glands, endocrine glands and exocrine glands. Endocrine glands make a substance that is secreted into the blood because this is some signaling molecule that is carried in the blood. It's defined as a hormone. So endocrine glands are always secreting a hormone, and we won't talk about them because they have their own chapter later in this semester. Then you have exocrine glands, which are secreting a substance either outside of the body or onto an internal surface. So that sometimes could be a signaling molecule, but it usually would not be. It would usually be something else. That would include sweat or sebum secreted onto the surface of your skin. That would include mucus secreted, onto your nose or onto your stomach. That would also include saliva, some digestive enzymes that are secreted from the pancreas into your intestines to help you digest, stuff like that. The pancreas can also secrete hormones into the blood, but it also has exocrine functions secreting enzymes into the intestine for digestion. The only specific type of gland you'll need to know for this class is the gland that makes mucus, that's a single-celled gland. Most glands have multiple cells composing the gland, but for your mucus glands, it's only one cell. The cells are called goblet cells, and you can see them like right here, right here, right here. So they're common to see in epithelial tissue, especially your simple columnar and pseudostratified columnar types of epithelium, not too much in the other types. And they're usually easy to recognize because it's usually like a pale blob within the tissue. So those goblet cells are just making mucus and then secreting it. Here's some other types of exocrine glands, the ones that are multicellular. They're generally going to be classified based on the shape and on how they branch. I'm actually not going to make you know these, so we won't go into it. Okay, and that's our epithelial tissues. So now we have the connective tissues and all the subtypes of connective tissues. Right, so different types of connective tissues have, of course, different functions. In general, connective tissues are always connecting things, holding things, supporting things, as the name implies. So pretty much any type of connective tissue would have those functions, but then certain types also have additional functions. So some connective tissues have a protective function. So bone is a type of connective tissue that protects your soft internal organs from damage. You also have fat. That's a type of connective tissue which provides shock absorption, so it also helps to protect your delicate internal organs from being damaged. You have functions in transport and immunity. That would mostly just be the blood. Blood is considered a type of connective tissue, and of course it's responsible for transporting all nutrients and waste products around your body, and signaling molecules like hormones as well. And immune cells will be traveling through the blood for immunity. Finally, you have functions in energy storage, specifically for fat. So one of the main roles of fat is going to be to store energy so that, when you can't access food, you don't just die right away. Right, so that's those would be the general functions of your connective tissue and the different types of connective tissue. Connective tissue is kind of more diverse than epithelial tissue is, so there's fewer common characteristics of connective tissue, but there are these two, that connective tissue is always mostly composed of extracellular matrix with few cells scattered throughout the tissue. Also, the extracellular matrix is going to be very important for the function of that tissue. It's going to be basically defining and creating the function of that tissue. So depending on what type of connective tissue we're talking about, we would have different components in the extracellular matrix in different proportions, and whatever that recipe is for the extracellular matrix, that's going to be underlying whatever function the tissue ends up having. And here's your categories of connective tissue. That's divided into proper connective tissues and specialized connective tissues. For your proper connective tissue types, you have loose connective tissue. Then you have dense connective tissue, which also has 3 subtypes. Irregular dense connective tissue, regular collagenous dense connective tissue, and regular elastic dense connective tissue. Then you have reticular tissue and adipose tissue, which is fat tissue. There's 2 subtypes of adipose tissue, white adipose and brown adipose tissues. So we'll go through each of those. Then you have your specialized connective tissue types. That includes cartilage, which also has 3 subtypes, which we will go through: hyaline cartilage, fibrous cartilage, and elastic cartilage. And then finally, the last 2 specialized subtypes of connective tissues are bone and blood. Each of those has its own chapter, so we won't really go into them here. Okay, so for our proper connective tissue types, you're going to have several types of cells that you would see in each of those subtypes, regardless of which subtype you're talking about, just sometimes in different proportions. So cells within connective tissue could be divided into resident cells that stay there all the time and migrant cells that come in and go out. The resident types of cells that you would find are fibroblasts, adipocytes, mast cells, and macrophages. Fibroblasts are the most common type of cell, and that's the cell type that's actually responsible for making the extracellular matrix. Right, so fibroblasts are actually basically making the connective tissue. Then you have adipocytes, which are fat cells. They store fat. They make and store fat. Right, so you of course see more of those cells in adipose tissue, but you would still see some of them in other types of proper connective tissue. You have mast cells, which is a type of immune cell. It's very large. Mast cells store chemicals called cytokines that promote inflammation. So if you have damage to the tissue, you would have your mast cells in the connective tissue be activated and start releasing their cytokines to start an immune response. Then you also have macrophages, another type of immune cell. Those are phagocytes, which means that they eat stuff. So macrophages are going to eat microbes if microbes happen to be around, but also just dead cells and cellular debris will be eaten by macrophages. Then your migrant cells would include more macrophages. Macrophages travel in the bloodstream in addition to residing in the tissue and can move in and out. So if you have an immune response that started with your mast cells releasing cytokines for inflammation, you're going to have macrophages from the bloodstream come to that area and migrate into that connective tissue to begin eating microbes. You also have other immune cells, many other types of immune cells, which could be migrant cells in connective tissue, which we're not going to talk about what specific types of immune cells those would be. You will learn about that in A&P 2. So our first proper subtype of proper connective tissue is loose connective tissue. That's also called areolar connective tissue. So if you're looking at maybe an older textbook or an older source, you would probably see it referred to as areolar. The extracellular matrix is mostly ground substance. You have all types of protein fibers suspended in that ground substance. You have fibroblasts, adipocytes, your immune cells, mast cells, and macrophages, and you have blood vessels. But lots and lots of ground substance. So in the picture here, like all these kind of pale areas, though that's all ground substance. and then it has fibers sort of moving through it. And also blood vessels. So the function of loose connective tissue is going to be to give support to whatever tissue is on top of it, which would be generally in epithelial tissue. It also functions in immunity and in supplying oxygen and other nutrients because it has all of those blood vessels. So you're going to find loose connective tissue most prominently beneath epithelial tissue. So as I had told you, you always have connective tissue beneath epithelial tissue, and that connective tissue has blood vessels because epithelial tissue never has blood vessels. And the specific type of connective tissue that you would have beneath epithelial tissue is always loose connective tissue, which has many blood vessels. Right, those are delivering oxygen, glucose, other nutrients, also bringing immune cells into the epithelial tissue if it's needed. Then you have dense connective tissue. You could also sometimes see that called fibrous connective tissue if you're looking at an older source. The extracellular matrix in dense connective tissue is always made mostly protein fibers. And then there's the three subtypes, which all have either a different type of fiber that's most common, or the fibers have a different arrangement. So we'll go through each one. First, you have dense irregular connective tissue. The main protein fiber in that extracellular matrix is collagen, so the tissue is mostly made of collagen fibers. You also have a little ground substance. It would be these white streaks here. That's ground substance, and then the pink stuff, that's your collagen. And then you also have little purple spots. Those are cells, mostly fibroblasts, to make all of that collagen. Right, so fibroblasts would be the main cell type in dense irregular tissue, and then the main fiber type and the main thing overall is collagen fibers. So since you know it's made mostly of collagen fibers, you could assume that it gives strength, that its role is to provide strength, which is correct. The collagen fibers in dense irregular connective tissue are arranged sort of irregularly, which is where the name comes from. So they run in all directions with no specific organization other than that they run in all directions. This allows the tissue to resist tension in all directions. So no matter what way you pull on dense irregular connective tissue, it's always going to resist that stretch. So you would use this type of tissue any place where you know you're going to have mechanical stress coming from many directions, or any place you can't predict where the stresses would be coming from. So you have in the dermis, the deeper layer of your skin, most of that is composed of dense irregular connective tissue. You can't predict what type of stress the skin would be exposed to, just that there will be a lot of it. You can also find that lining some of your organs, like the stomach and intestines, that would be subjected to a lot of stretch sometimes. That way the stomach doesn't just split open if you eat too much. Although it still will if you eat way too much. You can also find dense irregular connective tissue around your joints. Right, so as your joints are moving and bending, they're subjected to stress in many directions, well, depending on the joint, but the stresses can be coming in many directions and could be unpredictable. So dense irregular connective tissue will be providing strength there. Finally, scar tissue. Scar tissue is virtually always made of dense irregular connective tissue. Wherever you're putting a scar, it's usually not possible to predict where stresses would be coming from, so you just want to put the most general purpose strength tissue type that you have, which is dense irregular connective tissue. Okay. Next you have dense regular collagenous connective tissue. The main protein fiber there is also collagen. Again, it's pretty much just collagen with a tiny bit of ground substance and some fibroblasts. You don't have a lot of blood vessels in dense regular collagenous, nor do you have a lot in dense irregular, but you have particularly few in dense regular collagenous tissue. The main difference between dense regular collagenous and dense irregular is that the collagen fibers in dense regular collagenous all run in one direction. So as you can see in this tissue here, the fibers are all basically running horizontally, and they're all basically running together. Whereas in dense irregular tissue, they run in all directions. So since they're running all together in your dense regular collagenous tissue, that means that the tissue is extremely strong in one direction. So if you were to pull on this tissue like horizontally, it would resist that pull very well. It would be extremely difficult to apply enough force to actually stretch the tissue. But if you were to pull on it up and down, it might come apart kind of easily, because you don't have any protein fibers running in that direction. So you're going to find dense regular collagenous tissue anywhere you need extreme strength, but it's also possible to predict where the forces would be coming from, and they're coming only from one direction. So pretty much that means your tendons and your ligaments. Tendons connecting muscles to bone, ligaments connecting bones to each other. You can always predict generally how muscles will pull on bones, like what direction that pull would be coming in, how bones would pull on each other in what direction. So you can put your dense regular collagenous tissue there to provide huge amounts of strength in that one direction where stresses would be coming from. As the consequence of dense regular collagenous tissue having very few blood vessels is that if it is damaged, it's extremely slow to heal, because you're not having a lot of nutrients or oxygen come in, and you also can't really get stem cells or immune cells coming in very quickly because of that lack of blood vessels. That's why if you damage a tendon or a ligament, it would take so long to heal. Next you have dense regular elastic connective tissue. It's also sometimes called just elastic tissue if you're looking at an older source. So the main protein fiber in that extracellular matrix is the elastic fiber. There's also some collagen to provide just general strength, but it's mostly elastic fibers. So all of those kind of squiggly lines there you see, those are elastic fibers. The elastic fibers are running all in one direction. The collagen fibers that are there would be running in all directions. And the elastic fibers are going to allow that tissue to stretch and then bounce back to its original shape in just that one direction in which the elastic fibers are all running in. So you can find that type of tissue lining your largest blood vessels, prominently the aorta. As you have blood ejected into the aorta from the heart, the aorta is like experiencing a lot of strain there. It has to stretch to accommodate the new blood volume that's in there, and then it has to go back to its original shape. So you need elastic tissue, or dense regular elastic tissue, to help with that. You can also find dense regular elastic tissue composing a few ligaments in your body. For instance, ones that connect your vertebrae to each other. And there it would be allowing your back to twist and then kind of go back to its original alignment, so that you can turn your body around. Then you have reticular connective tissue. The main protein fiber in that extracellular matrix is the reticular fiber. Those fibers, of course, run in all directions to form something like a net. Reticular connective tissue is unusual amongst the proper connective tissue types in that it has many cells, a lot more cells than the other types of connective tissue have. You have a lot of fibroblasts in there to actually make the reticular fibers for the extracellular matrix, as well as all the other parts of the extracellular matrix. And you're usually going to have a lot of immune cells as well. It depends on where that tissue would be found. The role of it, since it's mostly composed of these reticular fibers, would be to give support to delicate structures and to trap stuff, for instance, microbes. So you're going to find that throughout pretty much the entirety of your lymphatic system. Wherever you have lymphatic tissue, your lymph nodes, your spleen, your other organs in the lymphatic system, that's generally made out of reticular tissue. Right, so those are the types of reticular tissue that would have many immune cells in them. Then you also have reticular tissue in the bone marrow. You have a lot of stem cells in your bone marrow, so the reticular tissue is kind of supporting them. You have reticular tissue in the liver, which is also filtering your blood, filtering all the blood that drains from your intestines and is containing, all the substances that were absorbed from your gut. And finally, basement membranes. The bottom layer of your basement membrane that was actually made by connective tissue consists of reticular connective tissue. That's why it's called the reticular layer, or the reticular lamina. Finally, adipose tissue. You have your two subtypes of that, starting with white adipose. White adipose is, of course, your most common type of adipose tissue. That contains fibroblasts, adipocytes, and collagen. So the main component of your extracellular matrix is collagen, and then you have a lot of fibroblasts and some adipocytes. So when adipose tissue starts off, it doesn't really have a lot of fat stored up, so all the adipocytes are really small, and you can see at that stage you would actually be able to see that the tissue is mostly extracellular matrix with some many fibroblasts and then some adipocytes. But as those adipocytes store fat, make and store fat, they get larger and larger. They swell up to contain like all that fat that they're storing. So then, once the tissue is sort of finished maturing, you would have just these adipose cells full of fat, and that swells the cell up. And you can see the little nucleus of the cell is pushed to the side, and then everything else that was in that tissue will be kind of pushed to the side and hidden from view by the massive balls of fat in the adipose cells. By the way, your adipocytes do not take fat from the blood and store it. They cannot do that. They take glucose from the blood and turn it into fat and store that. So if you want to get rid of some of that fat, you need to cut your glucose. It's not about how much fat you have in your diet. Places that you would find white adipose tissue would be underneath your skin, also in characteristic locations to deposit, fat. So around your abdomen, your hips, your butt, your thighs, and if you're a woman, your breasts. It would also be surrounding your heart and your abdominal organs, where it's helping provide some shock absorption to those organs. Under your skin, it's providing shock absorption. Everywhere that it exists is providing energy storage, which has been crucial for the survival of the mammalian lineage through multiple extinction events. But under the skin is also providing insulation to help keep your heat on the inside and help you maintain your body temperature. Right, then you have brown adipose tissue. That's more common in babies and very young children. You would lose it as an adult, so as an adult you have very little left over. The role of brown adipose tissue is primarily to give warmth. It's not really for storing energy, it's for giving warmth. And so it has a few differences from white adipose tissue that help it serve that function. So for your white adipose tissue, every fat cell has one single fat inclusion, one single ball of fat in the cell. In your brown adipose tissue, you have multiple small fat inclusions. You also have a lot of mitochondria, and you have a lot of blood vessels. So those multiple inclusions of fat gives a lot of surface area. So inside the cell, there's a lot of surface area of fat, of the fat that's being stored, which means that the cell can sort of break down that fat very quickly. The mitochondria are going to be metabolizing it to create ATP and glucose. And whenever the mitochondria do that, since that's a chemical reaction, it's going to be inefficient. Some of the energy involved with that reaction is going to be lost as heat, except it's not lost, it's used for the heat, right? So as these mitochondria are very rapidly metabolizing the fat, they're going to be giving off a lot of heat, and then that heat would enter the whole tissue surrounding the mitochondria, but also enter the blood vessels and be carried throughout the body in the blood. Right, so the function of that tissue then is to make warmth and transport it through the blood to the rest of the body. So babies and really young children don't have as much of an ability to maintain their body heat as an adult would have. They can't shiver very well, especially babies. Their muscles are weak. They don't have as much insulation, and they have a lot of surface area compared to their volume, which means they have a lot of places where they can lose heat, but not as many places to store it, basically. So the brown adipose tissue is like crucial to maintaining their body temperature, when they're so young. You can find that usually around the neck, the shoulders, and the back of babies or very young children. Okay, next we have the only type of specialized connective tissue that we'll talk about in detail, which is cartilage. You have 3 subtypes of cartilage, but all of them are strong and flexible. The extracellular matrix is going to have your ground substance components, your glycosaminoglycans, your proteoglycans, also collagen and elastic fibers, but the specific proportions of those components would differ depending on which subtype you're talking about. The prefix chondr always means cartilage, so if you ever see chondr, think cartilage. There's a couple of cell types that you see in cartilage, chondroblasts and chondrocytes. The chondra tells you that it is a cartilage cell. Chondroblasts are found on the outside of the cartilage. They actively divide and they make the extracellular matrix for the cartilage. As a chondroblast makes more and more cartilage all around it, it's going to end up kind of isolated in all the cartilage that it has made, and it will turn or mature into a chondrocyte, which is like a mature cartilage cell, but it's not active, and it's isolated within the cartilage in a little cavity that's called a lacuna, or plural would be lacunae. Lacuna would mean like lake. So those are the types of cells in cartilage. If you're looking at the interior of the cartilage, you'll see the lacunae, the little cavities, and they each have a chondrocyte inside. That's not an active cell. And then at the edge of the cartilage, you're going to see your chondroblasts that are actively dividing and actually making the cartilage. You don't have very many blood vessels in cartilage. Actually, within the cartilage, you have almost none. Around the outside of the cartilage is where you're going to have your blood vessels, which is also the same place that you have your active cartilage cells, your chondroblasts. That's all going to be surrounded by a membrane called the perichondrium, which is made of dense irregular connective tissue. So that is what's containing your blood vessels, and then between that membrane and the cartilage itself is where you're going to have your chondroblasts making the cartilage. Para means like around, like perimeter. So perichondrium is the thing that goes around the cartilage, which is the membrane that sheathes all cartilage. Once you go deeper into the cartilage, there's, as I said, like pretty much no blood vessels there, which means those cells are not getting oxygen if they're very deep into the cartilage. So the chondrocytes, as they get deeper into cartilage, as more and more cartilage is made, they're going to die. The innermost ones are always dead, and that also means that if you damage that cartilage, it's going to be extremely slow to heal because there's no blood vessels, and all the cells in there are dead anyways. Okay, your first subtype of cartilage is hyaline cartilage. That is the most common type. The extracellular matrix has a lot of ground substance in it, and then it has small bundles of collagen fibers. So that collagen running in all directions is giving it strength, and the high amount of ground substance is making it very smooth. Right, it's going to have a significant water content because of all those glycosaminoglycans and proteoglycans in there, which is going to, plump it up with water and helps to make it very smooth. So since it is so smooth, its role is going to be in reducing friction. So the places to find it most prominently would be covering joints. So wherever you have the surface of a bone that's actually going to touch another bone to make a joint, you're going to cover that surface with hyaline cartilage to reduce the friction between the bones as they move against each other. Also, you have a few joints that are made of hyaline cartilage. So you have joints between the ribs and the sternum made of hyaline cartilage. You can find it making your nose, giving your nose its shape, and also in your trachea. And then finally, the skeleton of a fetus is made of hyaline cartilage. And then as the baby is born and then grows, that cartilage is gradually replaced with bone tissue. Right, so that would be why, like, babies' bones are so weak. They're just made of cartilage, not actual bone tissue. Okay, then we have fibrous cartilage. You can also see that called fibrocartilage. The extracellular matrix has very little ground substance and a bunch of collagen, many collagen bundles. They're sort of mostly aligned, but not all the way aligned. So as you can see here, you've got some running this way, some running this way, some running this way, right? So they're not entirely aligned with each other, but they all are sort of running in, roughly the same direction. You don't have any running like up and down directly, for instance. You also have some elastic fibers in there. You're going to have actually some fibroblasts in that type of cartilage, which is unique for cartilage. And then, of course, you have to have your chondroblasts and chondrocytes, right? So let's see, here would be a chondrocyte. You can tell because it has its cavity, its lacuna. And then, right, this whole thing would be the lacuna. This type of cartilage is very strong because of all that collagen. It's also a little bit elastic from the elastic fibers, but since it has so little ground substance, it's not really smooth at all. It's not very common, but you can find it in fibrous joints, which are joints that do not move. So the best example is in the skull. You have the skull sutures joining the different skull bones together. Those joints are made of fibrous cartilage. The purpose of them is so that when a baby is born, the head isn't, or the brain isn't like the full adult size yet. So the brain has to be able to grow as the baby grows. So the skull has to get bigger around the brain to make room for it. And then only when the child's brain is like its final size is it possible for those joints to actually, harden and I guess be converted from hyaline cartilage to fibrous cartilage for the final joint. You also find fibrous cartilage in the intervertebral discs between the vertebrae in your spinal column. Right, the spinal cord is actually running through those vertebrae and through those intervertebral discs, so they need to be extremely strong. Right, you can't just have a continuous sheath of bone because you need to be able to bend and twist your back, or your motion would be incredibly limited. But you still need those spaces to be very strongly protected, because if your spinal cord is damaged, depending on where it's damaged, you'll be paralyzed and lose all functions beyond that point. The brain won't be able to communicate with the body beyond that point. Finally, you have articular discs in some joints that are made of fibrous cartilage and help distribute strain associated with the joint. So, for an example, here's a joint. Let's say it's your knee joint. This sheath here is dense irregular. Alright, so this is going to be dense irregular connective tissue. Then you have this blue stuff. That is actual hyaline cartilage lining the surfaces of the bones where they might touch each other to reduce friction. The sort of black space is actually space in the joint that would be filled with articular fluid, also to reduce friction. And then in the middle here, this white band, that is your fibrous cartilage articular disc. So that's going to distribute strain around the joint and distribute like sort of the weight. So for instance, you've got articular discs in your knee joint, which are called the menisci. You have a lateral and medial meniscus. Well, lateral meniscus and a medial meniscus. And yeah, those are just distributing the strain that falls on that joint, because that joint is bearing such a lot of weight, so that it's not just one part of those bones that's actually bearing the weight, it's the whole joint surface of the bone that is bearing the weight. Okay, finally we have elastic cartilage. That's very uncommon in the body. The extracellular matrix has, of course, many elastic fibers, as the name suggests, and it is elastic. There's also some ground substance in there, some collagen. You also have more cells than what's common to see in cartilage, right? So as you can see in the image, there's a lot of lacunae there, each having its own chondrocyte. Because of the sort of the combination between all of the elastic fibers and some amount of collagen fibers, the tissue is elastic, but not exactly in the way that other elastic tissues are elastic. So it doesn't exactly stretch and come back to its original shape so much as it vibrates. Right, so the role of elastic cartilage is going to be vibration. It provides vibration. There's not too many places in your body where you need vibration, but the two places are your ear and your voice box. So the cartilage of the outer ear is made of elastic cartilage, and as sounds come in, they're able to actually vibrate that cartilage. So the whole outer ear structure would actually be vibrating and helping to magnify sounds as they're funneled into the inner ear for, detection. So you're not aware of that vibration, but it does occur, and it helps you hear. Then in your voice box, right, of course your voice box needs to be able to vibrate to produce sounds. The voice box vibrates, and then that vibrates the air around the voice box, and then, or the air that's passing through the voice box, and vibrating air is sound. So if your voice box couldn't vibrate, it would be impossible to generate sound. So essentially, wherever you need a body structure to interact with sound in some way, which should be vibrations of the air, or vibrations of whatever material the sound is passing through, right, wherever you need that interaction with sound, you're going to have elastic cartilage to allow a structure to vibrate. Then you have bone, which we will briefly talk about. It's pretty much entirely composed of connective tissue. Well, bones are pretty much entirely composed of connective tissue. Right, at the ends of the bone where it's participating in a joint, you have the hyaline cartilage, which is lined with the perichondrium membrane, which is also made of connective tissue, dense irregular connective tissue. The bone itself, bone tissue, is a specialized connective tissue. It has a mineralized extracellular matrix, so the extracellular matrix has calcium and phosphate in it, making it very, very hard and resistant to bending forces. The bone is coated in the periosteum, which is also a membrane made out of dense irregular connective tissue that coats the bone. Ost means bone, so periosteum is the thing that is around the bone. Then inside of bones you have marrow, which is going to be connective tissue as well. If it's most of the marrow in an adult would be made of prominently adipocytes, so it would be like adipose tissue, white adipose tissue. And then the type of marrow that has stem cells in it, more common in children, is made of reticular connective tissue. But bone tissue itself has its own chapter, so we won't talk about it, really. Oh, well, I guess we'll talk a little bit about it just to point out that similar to how chondrocytes end up in little lacunae, bone cells also, called osteocytes for bone, also end up in little lacunae, such as right here. Yeah. Here you have another lacuna. Okay, then finally blood, which also is a specialized connective tissue with a highly specialized extracellular matrix. It's the only tissue in the body where the extracellular matrix is liquid. So blood plasma is the extracellular matrix of your blood. And then apart from that, you have red blood cells in there carrying oxygen and carbon dioxide, and you have white blood cells, which are immune cells. Oh, well, and then you have platelets, which, allow blood clotting. Okay, then you have muscle and nervous connective tissues, which we will briefly touch on. Again, they each have their own chapter, so we're going to keep it brief. For your muscle tissue, that is mostly all cells. The cells are called myocytes, myo meaning muscle, and of course cyte meaning cell, so muscle cells, myocytes. Myocytes are special because they are able to carry electrical signals, and then those electrical signals allow them to contract. Your myocytes, if we're talking about a skeletal muscle, the myocytes are extremely large sometimes. So they're usually going to run the length of the muscle. So for a long muscle, you would have an equally long cell. Those cells would have many nuclei in them. All right, and then if you strength train, instead of you're not going to gain more muscle cells. You always have the same number, unless some of them die, then you have less. But the muscle cells that you have are going to get thicker, and they're going to have more proteins in them that let them do the actual work of contraction. So each cell will be stronger and bigger. You have extremely little extracellular matrix in muscle tissue proper. Almost all of the extracellular matrix in a muscle is actually coming from connective tissue. It's not from muscle tissue. Muscle tissue is almost all cellular. So you don't need to know these layers at this point. You can learn them later when we get into muscles. But this is an individual muscle cell. And each of those cells is surrounded by loose connective tissue. So this stuff in here and around, that is loose connective tissue. That brings blood vessels and nutrients in to the muscle cells. And then you would have a number of those muscle cells all packed together, covered in another layer of connective tissue, this time dense irregular. That would be a muscle fascicle. Then if you get a lot of those fascicles together, you're going to have another layer of connective tissue. That would be also dense irregular. Then all the connective tissues are going to merge together at the end of the muscle to form a tendon. So that would be like the extracellular matrix in your muscle. It's not the muscle tissue itself, it's the various connective tissues that are surrounding the muscle cells. You have three basic types of muscle tissue: skeletal, cardiac, and smooth. Skeletal muscle is the type that you can choose to move. It's found in the muscular system of your body, which we will cover later in this semester. It is pictured at top here. So that's the type that has the super large cells with multiple nuclei. Then you have cardiac muscle tissue, which is only found in the heart, so only found in the cardiovascular system. That is not voluntary. You cannot choose to contract it. And those cells are connected with gap junctions, and they branch. And that gives them a highly synchronized contraction. So here would be one cardiac muscle cell, and you can see it branches a number of times, and then is connected here to the next cardiac muscle cell with gap junctions. Finally, you have smooth muscle that is also involuntary, and it's more varied. So it can do different types of contraction depending on where it is and what's needed. It's found in many places throughout the body. So you can find that type of tissue around your gut, well, lining your entire gastrointestinal tract, basically, where it contracts in a pattern known as peristalsis to move food down your throat and then through your intestines and through your stomach. Your blood vessels also, most of them have a coating of smooth muscle, so they can contract to raise your blood pressure and to control sort of where blood is flowing in your body. Sphincters are going to be made of smooth muscle. So for your urinary system, you actually have an involuntary smooth muscle sphincter and a voluntary skeletal muscle sphincter. Your smooth muscle that is found in sphincters can do this thing where it contracts and it locks in place, so it actually doesn't consume energy, but it remains contracted, and more. But we won't go into it in more detail at this point. Then you have nervous tissue, only found in the nervous system, of course. You have kind of a balance between extracellular matrix and cells in that type of tissue. It's a very specialized extracellular matrix. It's almost all ground substance, virtually no protein fibers at all, which means it's very weak. So that's why you're going to have your nervous tissue encased in other things, your brain encased in the skull, your spinal cord encased in your spinal column or your vertebral column, and the nerves that come out from that are sheathed in dense irregular connective tissue for protection. You also have a lot of signaling molecules in the extracellular matrix for nervous tissue. Neurons usually are not actually directly connected to each other, but they communicate between each other using these signaling molecules, which are released and then travel through the extracellular matrix from one cell to another cell. And finally, you have special cells in nervous tissue. You have neurons, which are the big cells here, right? So this would be a neuron. And you can see the processes coming off of it. Lots of processes that then branch. Right, so those are the main cells that we think of being in the nervous system. They're the ones that carry electrical signals and use those signals to process information. With all their processes included, they actually get extremely large. Some of those processes are super small. So for instance, in these parts of the tissue that might kind of look like that pink stuff is protein fibers, but it's actually not. It's actually tiny, tiny, tiny processes from the neurons themselves. So you can imagine that actually works out to a large amount, like a volume for those cells. Then you have last glial cells, various types of glial cells. Those are support cells basically. So all these little dots would be glial cells. They're way smaller. And there's a number of roles they play, support roles for the nervous system, which we will get into more detail with when we actually cover the nervous system next week. Okay. Finally, we will briefly touch on tissue repair, and then we'll be done. Okay, so you have two basic types of repair processes that tissues can undergo. You can either have regeneration or fibrosis. In regeneration, the wounded area or the area that was damaged is actually going to be filled in with new cells that are the same type of cell that was originally there. So the tissue is going to basically restore like it was new and get back its original function. In fibrosis, you have the wounded area filled in with scar tissue instead, which is dense irregular connective tissue for almost all types of scars. Right, so that is going to be very strong, but it's not functional. So, right, so you're getting the strength out of it, but losing whatever function was there. So, for instance, if you have a heart attack, right, if some of your heart muscle cells die, that would usually be replaced with scar tissue. which is good because it's very strong, and the heart needs to be strong. It has a lot of pressure inside of it with all the blood in there, but of course dense irregular connective tissue does not contract, so the heart, in terms of its contractile abilities, will be weaker permanently after a heart attack in which heart cells die. So there's a couple different factors that control which of these processes is going to happen after tissue is damaged. It depends on how frequently the cells in that tissue would divide normally. So for your stratified epithelia that have actively dividing cells at the bottom, like your stratified squamous epithelial tissues, those are likely to undergo regeneration because those cells under normal conditions are dividing very frequently. On the other hand, if you have a tissue type where the cells never divide, so for instance in muscle, then that's not very likely to undergo regeneration because those cells just don't divide. So where would you get new ones from? You would have to get it from like a stem cell. Also, how much blood the tissue gets, how much blood supply it has, is also going to help determine what process it undergoes. So for instance, for cartilage, you have like cartilage cells, chondroblasts, around the outside of that cartilage that do actively divide, so it could undergo regeneration, but cartilage gets very little blood flow, so it frequently will scar instead, or scar first and then take a long time to regenerate, and kind of not be as good as it started out being after it's done regenerating. Whereas when your skin heals, it will usually heal back just like it was. Right, so your skin also has a good blood supply, which helps. Right, so in any tissue where cells are dividing normally and you have good blood flow, that's likely to undergo regeneration. In any tissue where you have cells that do not divide normally and where you have not a lot of blood flow, that's likely to undergo fibrosis. So, epithelial tissues will frequently regenerate. Some of them have actively dividing cells at the bottom normally, and they usually will pretty much always have a good blood supply provided by the loose connective tissue underneath it. Connective tissue frequently undergoes regeneration, but with exceptions. So your dense, regular collagenous connective tissue and cartilage connective tissues don't have a lot of blood supply, so it's hard for them to undergo regeneration, but they can do it eventually. But for those types of tissue, it frequently would be actually okay for it to be replaced with scar tissue. You would have some reduction in strength, but not a significant loss of functionality beyond that. For muscle tissue, it's going to undergo fibrosis because those cells just don't divide. But for your skeletal muscle, those cells are extremely large in a large muscle, so if you just slash the cell at one point, it actually might not die. It might be able to just repair itself. So if the cell doesn't die, it can repair. Even if the cell does die, the other cells that are around it can get bigger, add more contractile units, and recover the strength that was lost. For your cardiac muscle tissue, that's generally just going to undergo fibrosis. It's not as easy for that tissue to recover its lost strength, although there is a small ability of the heart to regenerate. They have found a few actual heart cells that do divide, so it is technically possible, although it usually does undergo fibrosis. For smooth muscle, that tissue actually does regenerate. Those cells can divide and regenerate. Then for nervous tissue, neither. Instead of fibrosis or regeneration, you would have a glial scar formed, which is basically where glial cells are going to surround the damaged area and make a barrier. They're going to start laying down protein fibers to make a barrier. The point of that is to maintain the blood-brain barrier so that the nervous tissue is not exposed to other tissue types, because those other tissue types, the molecules they have, if they seep into the brain, it's going to interfere with the brain's functioning. So you have to have that barrier. It's very important. So the good thing about the barrier, the glial scar, is that it does maintain the blood-brain barrier. but the bad thing is that axons have a hard time getting by it. Right, so when neurons die, they're dead. They do not divide whatsoever, so they can't really be replaced. Although there is some evidence that maybe occasionally stem cells do travel from the bone marrow to form new neurons, but that's not a process that happens, frequently. So in general, when neurons die, like that's it, they're dead. But if, for instance, you just cut an axon, that neuron will probably not die. It will probably recover and just grow a new axon. Oh, the axon being the process, the large process that a neuron would send a message along, send a signal along. So it's going to receive messages on the dendrites and send them on the axon. So the new axon would try to find the same path that the old axon had taken. So it's going to try to retrace the same path in the nervous tissue that it had. But if it then runs into the glial scar, then of course it can't get past that scar. It would need to get around. So if that's a big scar, usually the axon would not be able to find its way around, although sometimes it can, particularly if it's more like on the edge of the scar. Then apart from that, your neurons that are left over that are not damaged but are kind of next to the damaged area, they can actually change their function. So they can take on some of the functions that were done by the neurons that are dead or are severed from, from what they had been connecting to. So you can regain functions that are lost, although initially, well, you're not going to have regeneration, and initially you're going to have, a glial scar form. Right, so here's an image of that. We won't talk about this process in detail. Right, here's some more images. So this would be an example of this right here would be like a glial scar in the spinal cord. Gosh. Right, this is a glial scar in the spinal cord. And then here you have a neuron whose axon was going through the spinal cord, and that axon got cut, but the neuron didn't die. So it's going to start sending its axon back down the way it had gone, and then the axon runs into the scar and can't get past. So here you have, right, so right here you have axons that were damaged. So you have like a cut in the spinal cord right here and a glial scar that formed, and then on either side axons are trying to get back through. But they can't do it, the glial scar is in the way. So this would be kind of one of the reasons why spinal cord injuries are so difficult to recover from. Usually you're not going to have too many neurons that die because it would be mostly axons that are cut, and those neurons will usually live, but the axons cannot get back through that glial scar. It's blocking their path. Although some of them may be able to somewhat if they're, kind of at the edges of the scar. So like, an axon might be able to sneak through if it's coming like right here, sneak through around the edge of that scar. I'm actually not going to tell you a story about my cat right now because we have spent many times on this. Okay, and here's a summary. So I'm not going to actually go through the summary with you, but you can go through it if you would like a summary on your own. Right, so that concludes our histology lecture. Thanks for bearing with me through the whole thing. I'm gonna post some questions over it on Blackboard, and yeah, that was histology.